Guys, so we're starting the day 2 video right now, and today we're going to basically be diving into microscopes, and we're going to be talking about microscopes a lot throughout this lesson, so pay attention. Alright, so first thing, we have two different types of microscopes. We have the light microscope, and then we have electron microscopes. We have different types of light microscopes. In this lesson, we are only going to be talking about the compound light microscope, but there are different types. And then we're going to be talking about the electron microscope, possibly tomorrow or the day after. And we have three different types of these. Just so you know, the electron microscopes are way more powerful than the light ones. The light microscopes are the ones that you've seen in the labs at school. They are very, very useful for when it comes to looking at cells. But if you want to get detail into the cell or to look inside the cell and see what could possibly be happening in there at such a tiny scale, you have to switch to an electron microscope. All right, so first thing, the compound light microscope uses visible light in order to see specimens. So we use light to see specimens. Pretty obvious stuff. It uses more than one lens to magnify an image. So I have a lens down here and then another lens up here, which allows me to magnify an image way, way, way larger than what it could have possibly been. So at the bottom, I have a bunch of lenses called my objective lenses. My objective lenses allow me to zoom in times 4, times 10, times 40, or times 100, right? So I have a bunch of them, and I can basically turn them and switch between them depending on how much I want to actually zoom in on an image. However, this is not really enough, right? You'd think like, okay, so if I zoom in on bacteria times 10, is that really going to make it big enough for me to see? The answer is obviously no. If you want to see the bacteria, you have to zoom in about 1,000 times or at least 750 times, right? So to be able to do that, we have another lens up here in the eyepiece. So we have lenses down here, we have a lens up here, and basically the lens in the eyepiece magnifies the image of what the objective lens is looking at, right? So basically, you can take the objective lens and the eyepiece and they will magnify each other and make the image even bigger than what it would have originally looked like. Okay? The only disadvantage to using this type of microscope, the advantage is you can use it to see living cells, you can see colored images, but you can only zoom in up to 1,000 times the actual size of the specimen that you're looking at, which sometimes really isn't enough. So if you want higher magnification, you have to switch to a different type of microscope. All right, so let's just go over some of the pieces of the microscope. So you have your eyepiece lens, which is in the eyepiece. It is usually times 10 or times 15, and this one you can't really change unless if you actually take it out and put a new one in. And then you have your nose piece. This nose piece holds onto your objective lenses and it allows you to turn the objective lenses. So let's say this is one objective lens. This could be times 100 zoom. This could be times 10 and this could be times 40 or times 40 and then times 10. If I wanted to zoom in 100 times, I would turn this until this piece is above my specimen. All right? And then I have my mirror or my light source. In this area, you will most likely find a light source. This is just a model of an older microscope. I just really liked it because it had good information on it. But this is most likely going to be a light source, and it's going to illuminate your sample so that you can actually see the sample because there needs to be light on it for you to be able to see it. Then there's going to be another piece. It may be found down here or up here, which is called the diaphragm. In the lab that you're going to be doing today, it's going to be down here. So as you turn it, it allows more or less light to be shined on your sample. Sometimes too much light being shined on your sample will make it hard to see, and obviously not enough light is also going to make it hard to see. So the diaphragm is going to allow you to adjust how much light is on your sample. All right, and then you have the clip. The clips are what hold on to your sample, and you have the stage. The stage is this. The stage can go up and down using the stage adjustment. So you have your stage, you put your thing on, and then you put your clips to hold your sample in place. And then we get to two very important pieces, the coarse and the fine adjustment, okay? So once I have the slide on my microscope, you'd be surprised at how difficult it is to actually find your sample. You're gonna sit there for a while messing around with it, trying to actually find it. So you're gonna use two different things, your coarse adjustment and your fine adjustment. Your coarse adjustment is the bigger knob, and sometimes it's found down here, like it's not always found up here. You guys will see it when you're working on your lab today. The coarse adjustment, basically you use it to find the basic outline of your bacteria. It's not going to help you get a whole bunch of detail, but it's going to at least help you find a fuzzy image of the bacteria. Once you have found the basic outline or that fuzzy image, you stop touching the coarse adjustment and you go into the fine adjustment. The fine adjustment will help you get a clearer image. So coarse adjustment helps you find the basic idea of the bacteria or of the cell that you're looking at. And then the fine adjustment helps you tune in and see a clearer image of that specimen that you are looking at. Okay? So today you're going to be doing this lab right here. I have already completed it. This is why I'm just going to go through it basically. You're going to go through the introduction. She's going to tell you what the whole lab report is actually about. Make sure you have your volume on so you can actually hear her talking.
We will use a light microscope with 10 times, 40 times, and 100 times lenses. The 100 times lens is an oil immersion lens. This microscope moves over the different parts.
So basically, you're going to go over different parts of the microscope, learn a little bit about them. See here, the diaphragm is at the bottom. The diaphragm helps you adjust how much light is in, and you're going to go through the entire lab as if you were actually using a real microscope. Believe it or not, even at school, we can't actually let you do this because it's such a complicated process and it would take a long time for you to actually learn how to use a microscope. All right, so you'll see that at a low magnification, you are going to use the diaphragm first to illuminate the whole thing. And then you're going to use possibly your coarse adjustment and your fine adjustment, and you're going to use your times 10 objective lens. And then when you want to move on, you might have to switch to another objective lens, which would be like your times 40. This would allow you to see the image with even more clarity and get an even larger image. And then finally, if you want to switch to this lens, the very last lens, which is your 100 times lens, at this point you have reached 1,000 times magnification and the whole thing is going to become a huge mess, right? The image is going to become super blurry, but the amazing thing is these microscopes actually have a way of dealing with that. If you're getting to 100 times and you have 10 times in your eyepiece, that means overall you have 1,000 times magnification. To make this work without it being very blurry, you have to use an oil to help reduce the amount of blurring of the image, okay? You'll learn about all of this, and there is actually a purpose of this lab, so you have to actually go through it to figure out what that is, because there's going to be questions about it in the quiz. All right, guys? And that's it. Now, you guys are going to go, you're going to do the lab report, and then you're going to do the quiz. And good luck. If anyone has any questions, I am always available. You can message me on Shobi.